Hey, everybody. All right, hang on, I'm trying to do yeah. All right, I'm gonna go through here and just mark off the attendance real quick. You guys know how this goes. Maybe it's gonna load. Corporate medicine. Just again. Felicity. I am sure Carlos will join us in a little bit. He tends to be a little bit late. That's okay. I'm not gonna worry about it. So, right. So I'm at home, in case you can't tell from my little background going on here. I usually like to work at work just because I get more work done then. But I was sick, man, all last night, and I'm being like kept alive by like Pepto-Bismol and like ginger tummy tea with electrolytes in it. That's pretty much what I'm rocking with right now. So, but yeah, but because I'm home, I put together like this really kind of elaborate, did a really great job on this presentation. I promise that I did, you guys. But, yeah, but because I am not at work, I apparently, even though I always save everything on my USB, I should have known that, of course, because I'm sick, that's why, but, yeah, I saved it at work. I saved it on my hard drive on my laptop, and I don't ever bring my laptop home because I save everything on my USB, which I didn't do. So I don't have my actual presentation, but it's fine. With what I did with my guys, and the last time you guys get to sort of benefit from the fact that like I've reaccumulated all my figures and everything like that. So it's pretty much the same. It's just less flashy. But yes. So today, we're going over exercise 2 for the lab. Before we get started, one of the questions that I had one of my students ask for, I don't know, just at the end of the lecture or whatever, asked about when the midterm is, which was like a big kind of question, but let me tell you. So we have a Unit 1 exam coming up immediately, pretty much on Tuesday. So Unit 1 is chapters 1, 2, 3, 4, 5, and 7. So skip 6. And believe me, there's a good reason. So those 6 chapters will be what's covered for the Unit 1 exam. And yes, it comes up this quick for the traditional class as well. It's just you guys get to have it condensed down in like pretty much half as much time, not less, because of it's an 8-week course. So Unit 1 exam is coming up. Now, Unit 1 is our bread and butter. Is if you're having a hard time with the information in Unit 1, because it's a lot of new information for that, it's pretty much the foundation that everything else is built off of for the rest of this. And it only gets more complicated from here. Unit 2 is going to cover genetics, literally hows and whys and wheres and whens of genetics, like DNA replication and DNA to RNA to protein, the central dogma of how protein is even manufactured in the cell by ribosomes, and mutations and different kinds of mutations, and how what tRNA does, and the amino acids, and genetic engineering and PCR and plasmids and transformation and all these things. And that's just the genetics. We also have metabolism, which is my absolute favorite chapter. If you haven't taken physiology yet, and you don't know much about metabolism, I can't wait to teach it to you, because it is like literally my favorite. Well, I don't get to. My video recording of me gets to, which kind of sucks, but I love metabolism. If you think you know why you breathe in oxygen and breathe out carbon dioxide, and you think it doesn't have to do with what you eat, then I hate to break it to you, it does. The oxygen molecules that are in your carbon dioxide that you breathe out did not come from the oxygen that you breathed in ever. Okay, so it's cool, and I get to tell you why you would suffocate if I would smother you with a pillow. So it's just the best. I think metabolism is the best. So that'll be in unit 2. So there's a lot of chemistry in that, as I'm sure you can imagine. Those are rough chapters. Usually I mean, not me, just want to be clear. Dr. Burkala, when he used to teach his course, used to say that unit 2 was the weed out unit. That the unit 2 exam was what would pick out the people who didn't have it in them to make it through microbiology. I don't think that's really true now. As long as you know what you're getting into with your unit 1 exam, you're actually prepared, and I don't treat you like babies for the unit 1 exam, then you'll be prepared for unit 2. Which I go out of my way to try to get you guys to really put your work in, without drowning you, hopefully. So, unit 1 exam is coming up. It's the first exam you're going to have in this course, and it's going to set the tone for everything. In my courses, not in Dr. B's courses, because Dr. B's, his worst test was always unit 2. My worst test is always unit 1, because people are not expecting the questions that I put on the test. And they're not meant to come out of left field. I do expect you guys to understand the information on your study sheets and not just regurgitate the definitions. There will be some questions that are regurgitation, I promise, but like there are also questions that are going to ask you basically critical thinking. There's a lot of critical thinking on my exams. A lot of people, I have had students though get mad at me. They'll be like, well, I never learned how to answer critical thinking. If you've ever solved a problem in your life, yes, you have. Like, don't start, like, don't start. Yes, you have done critical thinking. Are you joking? You have to use the knowledge and understanding of this material and apply it in a way to solve problems. If you cannot do that, you didn't learn the material, you just regurgitated. So if you can't do that, you will not do well on unit 1 test. And people often think that I'm exaggerating, but the average typically for my unit 1 exam is somewhere around 60%. It has been historically, not just for me, but also for Dr. Burkala. People just don't do well on the unit 1 exam. So that's why I do this. That's why I prepare you. I've noticed that people do better. It's not meant to scare you. And I'm not exaggerating it. I'm being honest with it. So I hope that that helps you guys get a better perspective on how you need to prepare. I will give you an example of a critical thinking question. I don't have it typed out here. I'm just giving you an example based on my memory. Let's say a guy goes and eats at a Chinese buffet, and he goes home and gets sick afterwards, right? A diarrhea, okay? He has GI problems. And it came on pretty quick after he ate it. We'll say within 6 to 12 hours after he ate whatever the food was. And this ends up happening to a whole bunch of people that ate at the same Chinese restaurant. So an epidemiology team is set out from the health department to go figure out what's going on at this place. And they do testing, and they find endospore containing bacteria in the rice. Okay, not uncommon, but in the fried rice, there's endospore containing bacteria. Now, if you've read your chapter 4, you should already know where this is going, but here's your choices for what you think caused. That's it. That's the information you get. Okay, I'm gonna give you 4 choices. Was it Bacillus? I should write it down. I'm gonna write it down in the chat so you guys can actually follow along what I'm saying, because you don't know words. Mine. Was it Bacillus cereus, or was it Staphylococcus aureus, or was it Escherichia coli, which E. coli, right? Or was it, I'm going to send it in a second, Salmonella enterica. All right, so there's your 4 choices. Now, you don't have to answer right now, but I do want you to start thinking about this. If you haven't started studying at all for this, these are the questions you're going to get faced with. Like, this is just like how a little bit of it is like, I expect you to be able to apply the information that you're learning, and to questions like this. So guy goes to Chinese place, gets fried rice, gets sick. Okay. Everybody that got sick ate the fried rice, and the fried rice has endospore forming bacteria. Okay, so that's our big key. Why would I include that if that, right? So that there's something there. Endospore bacteria. Okay. Okay. So if you haven't read chapter 4, this is where you would get your answer from, right? We know it has endospore forming bacteria and that's causing diarrhea. These are your choices. 99% of my students will choose salmonella for this, probably because they don't study. And they know diarrhea is caused by salmonella. Well, all of these cause diarrhea. Hate to break it to you. 100% of them cause diarrhea. So you're not winning with that one. Yeah, so that's usually the choice. Everybody just goes with salmonella and they think diarrhea with that, right? It is incorrect. The correct answer is Bacillus cereus, and this is why. There are only 2 genuses, and if you have been studying, again, chapter 1 tells us that when you name these organisms, Bacillus cereus, Salmonella enterica, Escherichia coli, the first name is the genus, the second name is the specific epithet that gives you the species as a whole. This genus, Bacillus, Staphylococcus, Escherichia, Salmonella, the first part. Endospore forming bacteria, there are only 2 genuses of bacteria ever in 100% that form endospores, okay? That is Bacillus species and Clostridium species. Some Clostridium that you might be familiar with. Clostridium tetani, Clostridium botulinum, Clostridium difficile, Clostridium perfringens. That's gas gangrene, by the way. It also causes food poisoning, but it's not on the list, is it? So Bacillus and Clostridium should be what you're thinking when you see endospore bacteria. Which of those has Bacillus or Clostridium? It's that. You see how I just made that easy when you thought it was complicated? That's how it should be. If you know your shit, then you should be able to answer these questions. You should be able to know that, okay, endospores. Oh yeah, that's Bacillus and Clostridium. Yeah, that, and even if you can't remember specifically if it's Bacillus or Clostridium and you have the idea that you know you had rehearsed that in your flashcards, you'll recognize it in the choices. It's always all multiple choice. So if you study it and you really learned that material, you'll be fine, okay? But expect that. I will not disappoint, believe me. I seem nice and fun and bubbly until it comes down to you having to learn the material and I say, I put all this effort in and you didn't. What are you doing? Yeah, you gotta put in the effort too, man. It's not one-sided. A lot of people think it is. It's not show up and get the grade. Yeah, so there you go. On the 11th, we have an exam. It's gonna be unit 1, 1 through 5, and 7 are the chapters. It's the bread and butter. You get through that and you're comfortable with that, you'll be set up pretty good for the rest of what's coming up. That's what I think. If you do poorly on unit 1, because like I said, that average usually is pretty low. It's usually the worst test that I have, and it's usually about 60 average. That's what unit 2 is kind of for. I'm making it a comprehensive. I don't usually do that. But if you do better on unit 2, which includes unit 1 again, then I will give you that score for your unit 1 instead of your original unit 1. So you have a little bit of something to fall back on if you need to. But unit 2 is difficult, so don't bet on it, okay? Got it. All right. Getting on to the actual stuff. Exercise 2. So my stomach is really bothering me. Give me just a second. Ooh, okay. Calling morphology. So I didn't have my slides, so we're gonna go through this anyways. We have all the information we need, so it's not a big deal. I just made a sheet for you that I'll be posting with all of this that has like the pictures and stuff that we're gonna be using in the talk. So we'll be set. So this is your actual worksheet that would be going on with the lab that you would be turning in the back of it, right? So this would be your dry erase worksheet. So we'll start with colony morphology. Colony morphology is talking about the colonies, whatever you see growing on the plate, and what they look like. Morphology, like if you think morph, morphing and shapes and everything like that. So the colony, shapes, and the study of that is ology, okay? Break it down. So when you're looking at the colonies on your plates, you're going to describe them because those can be defining characteristics that can help you separate out types of bacteria. And I'll show you because we're going to go over that in our questions at the end. But here's some examples when I bring these up. Because these will be dealing with, I hate that this is in the way right here. Okay. All right, so if I were to say this was your plate that you got for your result for the samples that we were going to look at, which we aren't going to because we don't have it, because we don't have a lab. But if this was your plate, what you were looking at, your colony morphology would be to describe these like little kind of off-white, round, smooth, they're opaque, that sort of stuff, yeah? Whereas if somebody else had this plate, you see the difference already? Like you can look at that and already tell that's the bacteria that are that color. This is a bacterium called Serratia marcescens, and it's notoriously red like that. You ever had like on your sink when you don't clean it for a long time, that reddish gunky growth that you get, and you know it's like bacteria? Serratia marcescens, very red color, just naturally to the bacteria. And found pretty much everywhere. It's a ubiquitous type of bacterium, but you can already see the difference between this and this. And we see this sort of golden yellowish color to these. That's kind of what can give the name to some bacteria. Staphylococcus aureus. That aureus in its name refers to the golden yellow color of the colonies. Now, a lot of them can have that color and look that way, but it's stuck with that one. I don't know what this is if they're growing this plate. It could be a lot of different things. A lot of them look pretty similar, but sometimes they don't. Sometimes they're unique like the other one, right? So that's why we focus on it. Use all of this information to determine what your organism is. So whatever. So the shape of the colony, so those little dots that were on there, the actual shape of that dot, is it round? Is it irregularly shaped? Is it punctiform? That's the little dots, like a whole bunch of tiny little dots all over. The margin, that's the whole edge of the entire thing, the edge around the edge base, as well as over the whole top of the whole thing. So it's margin. It's like it's covering, yeah. Is this margin entire? Is it smooth? So it has this nice smoothness like we saw in the picture that I was just showing here. I mean, clearly these, if you were to look at them, probably they're going to be smooth and nice all over the whole top surface of that thing. And so on and so forth. When we look at lobate, we are looking at a surface that has like basically lobes, kind of like the edges of a flower all around the edges of it. We have filamentous, so filaments, they'll be going off like this, like little hairs going up to the side. And then we have rhizoid. So those are branching filaments. They look like they're branching off like branches of a tree, literally. So it's the difference between those straight shoot-offs, like hairs, or branching offs, like a tree, rhizoid. The surface of it, is it smooth, rough, or wrinkled? It's pretty straightforward when you look at it. Texture, is it moist when it is manipulated? Is it mucousy and sticky? Is it butyrous like butter? Is it umbonate? Sorry, that's elevation. I just looked right at the term and I just moved on. Is it dry or shiny or dull? The texture of the actual, colonies. Then we have the elevation. When you're looking at like the flat surface of the auger, what does it look like from that angle? Is it flat? Is it slightly raised? Does it have a curve to the top of it? Does it have a very curved, like almost like a big hill? That's pulvinate. Umbonate is like a fried egg. So you have like some flat growth and some raising in the middle. It can be a little bit off and irregular too. And then we have crateriform or crateriform, however you want to say it. I'm not picky about spellings, okay, as long as you get the concepts. Which is like volcano-like, so I'll have a dip in the center. It can be smooth or it can have like rough edges and actually look like a volcano. But yeah, so that is the idea with colony morphology. And then I've got this here as a little picture for reference. So if we look at this, filamentous is going out, rhizoid, we've got some branching going off on that. And all that. So these are some examples of what you would see. Again, I'm uploading this document for you guys when I put it in that discussions, if you guys ever want to reference that, okay? This is also in your lab manual, not this exact picture, but similar stuff in your lab manual for this, which is also a good place to reference it. Probably better because it has explanations alongside of it. But I needed something quick. So next we're going to talk about the microscopes. So great, so you grew your plate. Yeah, nice. You grew your plate and you've got these colonies, and now you want to look at your bacteria in the microscope. So first thing is to be familiar with your microscope. And all the little parts on it. So we'll look at this little shape, this little guy here. Let's make him a little bigger. So if you look in a microscope here, well, we'll start with the base. So it's the bottom, the arm, it's like the spine of the microscope that you'll grab the spine, the arm, put your hand under the base to carry it, just to be sure it's nice and secure so you're not going to drop it anywhere because it's expensive. The light source, which is literally like the light bulb where the light is coming from, it's at on the base down here. They also call it the illuminator here. Doesn't matter, same thing. The condenser is the thing underneath this flat part here. So this flat part, first of all, is called the stage. Think of it as being like a stage. That's where you're going to put your slide. That's where the show is going to be. So under the stage, we have the condenser to help focus the light from the light source onto your specimen. Okay. So that we can see it, you know. You can control the light going through the condenser via the iris diaphragm, or just the diaphragm. That's fine. It controls how much light passes through the condenser and onto your specimen. Your specimen will be sitting on this little slide right up above that. Okay, the light shines right through the area where your specimen is on the slide. The clips will hold that slide into place. All right, so we've got it all on the stage and ready to go. If your slide, when you put it on the stage, isn't in the right spot and you need it to be moved up or more over in order to be above that light, you do not push the slide around. You do not push the stage around. You use the little knobs on the side, out to the side on the right down here. I don't have it on our microscope parts list because it's not something you would get tested over, but it is known as the stage controls. One knob makes it go forward and back, and the other knob makes it go side to side. Okay. Use that, I promise. All right, then we have the light passing through your specimen and into a lens. The lens that it hits first is known as the objective lens. We have 4 lenses on our microscope: 4x, 10x, 40x, and 100x. And that's it. So you always want to start on the 4x. You want to be sure that when you're first beginning, your stage is all the way down. You don't control up and down with those stage controls. You control up and down with the focuses. And the coarse focus does big movements of up and down stage. And the fine focus does little movements of up and down stage. And yeah, that literally is how you're focusing it, by moving your slide further or closer from your lens. A lot of people think that that affects the lens that you're actually looking at. It does not. It affects how the stage is seated immediately in relation to your lens. So if you need to move the stage up or down, you will use the coarse or fine adjustments. So on the 4x, you always start out with the lowest. That was louder than I expected that to be. Yeah, so you'll start out with the lowest objective lens, 4x, right? Always, and you will get an image in focus there using your coarse focus first and bring it just real, like you can use big movements. It's okay. You're never going to get close to that lens and crack it or anything like that. So you're fine on the 4x. So use the coarse focus on the 4x. And then when you see it, get it into fine, perfect, fine-tuned focus using the fine focus knob on the outside. It's the little one. Okay. Once it's good, you will switch over, just flip it around turn it around using the nose piece. The nose piece is the part that turns and switches between the lenses. Then you'll go to the 10x is the next one. You'll just use the fine focus there. You need to use the coarse. Then you'll switch to the 40. Once you found it clear on the 10, you'll switch to the 40 because they should be very closely in focus to one another. That's what we have them calibrated for. So then you'll use your fine focus again to get it very as close as you can. And then once you're ready to go to your 100x, and most of our work will be done at the 100, okay? The 100 always uses immersion oil. And it is the only lens that uses immersion oil ever, okay? But if you're using that 100, you don't need to be switching to it without putting immersion oil down first. So you will have your 40. It was clear on the 40. You will kind of switch it over, leave some space before you go over all the way, put a nice healthy drop about the size of the light source onto your slide of the immersion oil, and then just switch the 100 over onto it. And then it will sort of form that sort of, we think of water tension, but it's oil, touching the lens and to the glass. That is because as light travels through the glass and it's going through the glass of your slide and it hits, if it was air, there's major density between air and glass. So it causes the light rays to scatter. Well, that's called refraction. And so instead we have the oil there and it's a lot more similar to the glass. So it prevents the light rays from scattering. So we can get a nice, tight, clear image when we're zoomed in that far. So that's why we do that, to reduce the light scatter. So great. So we added our oil and now we're looking at the 100 and you'll use the fine focus. You should not be touching the coarse focus ever past the 4x, okay? And always work through the lenses, please. It's just a lot easier if you do it that way until you become like just total expert on finding things in the microscope. There are easier ways to switch over to the 100x, but I don't want you doing that until we've done many a microscope, okay? You risk, if you use that incorrectly, you risk cracking the lenses on the 40x or the 100x. It's extremely expensive to get that fixed, or by breaking the mechanisms of how they sort of have this like bounciness to them. We don't want to break that, and we don't want to break your glass slides thereby causing, you know, safety hazards and stuff like that. So just follow the rules. But that is the basic setup of the microscope. Our microscopes in our lab, in our eyepiece, which is what you're looking through, they have a lens in them that also help magnify. It's a 10x lens. So whatever lens you're on your objective lens on the nose piece that you're turning it around, right, that sees your image first. So whatever your specimen is, light goes through it, right, into that lens in the objective lens. And that is called the real image because it came straight from your specimen, okay? It'll bounce through some microscope, it'll bounce through the mirrors within the microscope. And I get so distracted when I hear noise. Sorry, it's the ADHD. Okay, we'll bounce through the mirrors in the microscope and go out the lenses of the ocular lenses. That's a 10x magnification, like I said. So we got a 40x, excuse me, at the beginning, 4x, then mirrors, and then another lens, 10x. That'll create the virtual image because we already went through 2 lenses and we had mirrors in the middle. Oh my gosh, it's not the real image anymore. It's the virtual image that you're seeing with your eyeballs, okay? Went through all this stuff to get there. So in order to figure out the whole total magnification, all you do is multiply. And since our eyepieces are always going to be 10x in our lab, it's easy multiplication. It's basically like 10 times whatever your objective lens is. So if you're using a 4x, 10 times, it's adding a 0 on, so 400. If you're using the 10x, that would be 100 total. If you're using the 40x, that's 400 total. And if you're using the 100x objective lens, that would be a total of 1000 times magnification. So when you're actually looking at your bacteria, you're actually looking at what hits your eyeball is 1000 times bigger than what it really is in real life. So that's how that magnification is calculated. And it's pretty good. All right. So let's say first, before we're going on to these cell shapes and whatever. So now you're making your slide. You're making your actual slide of your specimen, and we had the growth on your plate, and you're ready to pick one of these columns, the individual ones that aren't touching anybody else, and put it onto your slide. This is not hard. If you're making it from broth, like a liquid, and you just screw it up, the broth will turn cloudy whenever it has the growth in it. All you'll do is take your sterile loop and get a little bit of that broth, you know, like I said, you're going to look like you're going to blow a bubble with a little film. And just touch it onto a glass slide and just spread it out. And about a dime-sized area, and maybe nickel, and spread it out and let it air dry. And the more you spread it, the more faster it's going to air dry, I'm just saying. So you'll let it air dry, and good. And then you're going to heat fix it, which I'll get to. And then you can stain it and put it under the microscope. It's easy with the broth, you just spread it out and let it air dry. If you're picking one of these colonies though, it's not going to spread. really very well. I mean, it kind of can, but it's way easier to mix it with just a touch of water. And I mean, just a touch. Don't use your little squirt bottle and squirt a whole drop onto your glass. That's way too much, I promise. It's gonna take forever to dry. So what I do is take that squirt bottle and squirt it onto my loop on my little sterilized loop, and just in that little loop area, get about as much as I would have of the broth. Just a little film of water, and just touch it to my glass. Sterilize it. So just a touch of water on there. Sterilize my loop, get my colony, pick my colony off of my plate, just a touch of it, enough that you can see that it's there. Mix it in with the water and then spread that out. Okay, dries way faster if you put less water, but you need a little bit to help it spread out evenly. You want a nice cloudy film over the top of the glass slide. You want a nice cloudy film. You don't want it to be completely see-through. If it's nice, it's really chunky and you can't even see through it on the glass, the light can't travel through it and you won't be able to see anything in the microscope. It'll be too much bacteria piled up on one another and you won't get any detail. I can usually make most things work. There's been very few slides that I have to tell people to remake them, but just you spread it out nice and thin, and that's way easier for me to work with. It's way easier for you to work with, and it'll be much more user-friendly. So you let it air dry, and then we're going to heat fix it. So what was heat fixing? Heat fixing is one of probably the most important things that you're going to do with your slides. For most of them, we have one experiment where we are going to be using air-dried slides that will not be heat fixed, and that is the capsule stain, and we'll talk about why when we get there. But everything else we're pretty much heat fixing. So after you've air-dried your smear on your glass, you are going to take your slide. So you got your whole slide, and you got your air-dried crap on it, and you've got your Bunsen burner flaming over here, right? And what I like to do is take it through the top of the flame, just right kind of up here through the flame itself. I use a little tongs to hold it. We have them in our drawers in the lab, so you don't burn yourself. And I just go like in and out. So I'm actually pretend I'm going through the flame, please. In the flame and out the flame, kind of slowly. I do that like 3, maybe 4 times. I have a pretty good feel for when I think it's done, because I've done so many of them. But 3 or 4 times through that flame slowly. Don't let it sit there because it will burn your sample, and that will just ruin the structures of everything that's there. And you don't put it in when it's still wet, because if you put it in when it's wet, it's going to boil the sample and it's going to pretty much ruin it just the same way as if you burned it. So you don't want to do those things. You want it air dried nice, and then you can pass it slowly through the flame. You want to keep it moving. You don't want to stop. All right. That's how you create the heat fix, but what is the point of the heat fix? Well, the heat fix is going to kill the bacteria, number 1. Number 2, it's going to help adhere your sample to the slide. And when we do not heat fix, which we won't in the capsule stain, you have to be careful with your prep to be sure that you're not going to rinse it off, which you have to do special procedure for capsule stain because of that. Anyways, yeah, so you kill it, you adhere it to the surface, and it also helps to preserve the cellular structure. As long as you're not burning it or boiling it, you can preserve the structure of everything going on with those cells at the moment that you're doing this. It also stops the metabolic processes if you were trying to look at stuff like that and effects of that. So that is why we will heat fix our slides. So there's a lot of benefit to it. So once we have our slide heat fixed appropriately, we will let it cool before we move on, usually, because if it's super, super hot and you add your stain on there, it will boil the stain. So you want to be sure it's cool enough. It's okay if it steams a little. That's actually not so uncommon because I'm pretty impatient, but yeah, so you would let it cool appropriately. If you can touch it with your hands and it's not hurting you, then you're fine to put your stain on. And if we're doing a simple stain, so the first thing we learn about here is our simple stains. All it is to add some color to your cells, and it's the cell parts that gives contrast so that you can see it better. Because if you don't stain your cells, they're basically clear, and it is really hard to see them. That's kind of what the motility wet mount is about. We're not staining those cells because they're alive. We're doing a wet mount. We're not doing heat fixing or drying or anything. They're alive. So we're trying to see if they're swimming around and stuff, but you can't stain them. So you have to find swimming, moving around cells that are clear, which is so hard, but it's one of my favorite. It's so fun when you see them just moving around and everything. So I'm hoping that we get to do that one, but and it is what it is, right? But yeah, so here we have killed cells, so we're going to stain them to create some contrast to help see the cells and the parts inside of them. So simple stain is easy. You're just going to do your heat fixed slide, and then you're going to put one simple stain on there, and let it sit for like 1 minute, and then rinse it off with deionized water, blot it, and look at it in a microscope. It's just that simple. Maybe that sounds like, oh, that's work though. When you get to know your gram stains and things like that, you'll realize how easy the simple stain is. But yeah, I use methylene blue typically. It's one stain that I use. It's not like you have one that you have to require to do. There's a few you can choose from. So I choose methylene blue typically. And you just put it on for a minute and then dump off the excess and then rinse it off with DI water. And we blot it with paper towels. That's what we do. And let it kind of air dry before you look at it under the microscope. Again, just to get contrast. So on your, let's say this was your simple stain that you did in your lab, and you'll have your questions that you'll answer on the back of the sheet afterwards, but you're going to pretend like this was what you had and what you might see. So you can see your cells and everything like that. So now you might be like looking at this and thinking like, oh yeah, cool. So that's what bacteria look like. That's what some of the bacteria look like, but how you might distinguish them. So let me actually show you something. This is fine. Purple, great. I don't know why it thinks that's what I'm asking. If you were to do a simple stain with methylene blue with Staphylococcus aureus, I don't know why I'm doing that, it would look like this. So you see that that is different than what I'm showing you with what your result would have been, quote unquote, with this. Potentially, this is your fake result, obviously, but you guys would have different results for your tables, ideally. But yeah, you see the difference there? You see the way that they're bunched up on the left? You see the way that they are round instead of that long rod shape? And you see how the rods, they're not bunching up like that. You have a few that are stuck end to end, maybe a couple here or there stuck end to end, but you don't see like bunches up like grapes almost, right? So those are defining characteristics of how you can identify some organisms. So Staphylococcus aureus, I told you that their colonies on the plate have that golden yellow glow to them. That's why the aureus, but the Staphylococcus part, now there's a different thing. So there are basically, when you talk about bacteria and what you see in the microscope, especially on a simple stain, but you can see this in any stain. The cell shape and the cell arrangement are different things. So what each cell individually is shaped like versus how those cells arrange with one another. So arrangements or grouping. The two shapes, there's a bunch of shapes, y'all. The two shapes that I really want you to be familiar with in the lab, and the only two that I'm ever going to ask you about on the exam. Cockeye or coccus. For single is coccus, cockeye is multiple. That's round, and bacilli. Bacilli are rods, okay? So yeah, so you remember how I talked to you guys at the beginning about the question about the endospore form of bacteria that cause diarrhea. And that was Bacillus cereus in that example. So bacillus, so you already know something about that bacteria right there. You know that it is a rod-shaped bacteria immediately because it's in its name. Staphylococcus aureus. What's the shape of those cells? Coccus. Staphylococcus aureus. So you know a lot about that bacteria right there. It's got the golden colonies and it's got the round cells. You actually know even more than that. Because when you move on down to those arrangements, you'll see that staphylo is an arrangement. Staphylo means irregular clusters or grape-like clusters. Think of them as grape-like, okay? They'll kind of huddle together in big old piles. And that's very characteristic of Staphylococcus aureus. So staphylo, we have those clusters. Coccus, the round cells. Aureus, the colonies have that golden color to them. You learned a lot about that bacteria just in the name. Not all of them are named like that. Of course, they have the weird names sometimes, but some of them are this, like Escherichia doesn't tell you anything about anything to do with it. Which, by the way, Escherichia coli are rod-shaped bacteria, but it's not in the name, right? So, and that's okay. It's usually going to be in the genus that you see the shape and arrangement, if it does have the shape and arrangement in the name. So Staphylococcus aureus versus Streptococcus pyogenes. Streptococcus pyogenes is the cause of strep throat, for example, and it isn't going to be in the clusters. It's not staphylo, it's strepto. The way I remember the difference, strepto to me is like strips. Okay, strips like chains, and they're chains of the cells all hooked together like that. You can have chains of the rods, as it shows us next to that, over here on this side. Streptobacilli they can link up in chains like that. And then the other big groupings, you have Sarcina and Tetrad, which I'm pretty much never going to test you over, but they exist and they are present in some characteristic bacteria. And then Palisades for the bacilli, which we don't typically see them form this way in the lab with the Palisades, but you can see it looks sort of like fences. So they're almost like up next to each other, but they're attached by their ends still. It's like you had like the sausages all linked together and then the links on end to end, but then pushed them all up upright. It's sort of like that. But yeah, they just stuck that way because they divided that way. And so you can see here diplobacilli, there's just two of them stuck together. I think when you look at this picture down here, you can see a couple of like 1 and 2. It looks like there's 2 stuck together here. There's 2 stuck together maybe over here, and that sort of stuff. So yeah, so you can sometimes see that going on. Compare that to Neisseria meningititis. When you look at a stain of that, I mean, obviously this isn't a typical simple stain, but look at them. Diplococci, they're a little two. This is one, and this is one. This is one. Look at little butts, like just floating around, like a little butt here and a butt here. But these are actually 2 cells that are stuck together. They are diplococci. They're actually round cells that are just stuck together really tight. Very characteristic presentation of all the Neisseria species. So if you're looking at Neisseria meningititis that causes the worst kind of meningitis that you can have, the one with the rash, that's what that would look like. And then they're gram negative. So when we learn about the gram stain, they stain negative. So they have a pink or red stain to them because they're gram negative. So very defining characteristic there when you're looking at the shapes of them as well. So yeah, especially if you're comparing them. Well, I just feel like I lost my train of thought. Anyway, so cell structure, cell shape, cocci or bacillus, and cell arrangements, strepto, staphylo, diplo, palisades. Those are the biggies. Okay. Cool. So that's the gist of how we would analyze our results that we would see here. So we're looking at our samples if we had gotten any. So when you guys are going through, that's pretty much all I needed to teach about it. But when you're going through this, so I didn't fill this one out for you. You're going to have to do this one on your own this time, right? So you can get these worksheets from Canvas. Again, if you go to modules, go to the bottom where it has lab worksheets and documents. It's the second one down there. It's in lab participation worksheets. Or you can pull it off of Top Hat. Top Hat has it as well. They're individual on Top Hat. So for each exercise. And you should be able to still reference those even after the thing was due. So you can use that. And then fill these out. Number 1, and I'll post this BS whenever I get to it. But it will say question 1, this is your plate. So it'll say using appropriate terms, describe bacterial growth on your street plate. Number 2, did anyone's plate look different? Why might that be? This is number 2. This one looked different. Why might that look different? Describe it as different bacteria. That's fine. And if you want to get into the detail of why bacteria look different, then that's fine too. I'm not going to stop you, but really just appreciating the fact that like not all bacteria look the same. Really, it's that simple, but making you think about it in a different way. Question 3 is the reason why I ask it in weird ways sometimes, just makes you think about it differently. Number 3, what shapes or arrangements would you describe your simple stain, which is going to be this image here, as exhibiting? So then you would pick your cell shapes, you would pick your arrangements. Oh, I should point out too, with Coccobacilli, because eventually we will see Coccobacilli to real bacteria. Some bacteria, like Corynebacterium, if they don't have this club weird shape, the Corynebacterium will have this Coccobacillus, and a lot of people will confuse it with coccus because it'll have such a rounded shape. So sometimes there's gray area. That's what my point is here. There's gray area. It's not always defined. So keep that in mind. But yeah, we're not doing the grand rounds. So you can just X through that if you want to. If you guys want to print this out and write on it by hand and then take a picture of it, great. If you want to use your tablets and your fancy little programs to like write on it and then send that, great. If you want to just make a whole other document on the side, like a Word document or a notes document, I don't care what, and just put like number 1 and then describe, number 2 and then describe it, and then number 3 and then answer it, and then that's all you want to send me. It's just written out and no fancy, not even a picture of this or whatever. It's fine, just as long as you have all your information there. I can see that you're getting the concepts. That's the idea. Okay, so what do we need to do to get it there? Next one's Grimstein. Okay, I feel like that covered everything. I'm just trying to think if I missed something. I have and I even have it on this. This is the document I'm going to be uploading. It has all of this stuff on here. And then I have an old video of this exercise that I posted where I was probably exceptionally frustrated. Just if you do go watch it, but it is on YouTube, and that is the link to it that I have there. And I'm going to post all that, so no worries. But if you want to watch this one, it is the whole lab intro stuff, and I walk through the lab basically. It's from years ago on a snow day. It was like iced out, and like I'm not gonna lie, I went into the lab, our micro lab. It was pretty bad weather, but I didn't want to get behind, have people have to worry about getting behind on this stuff because we'd had several snow days. And I just drove on to campus. I have a 4-wheel drive, so I just went into campus and knocked out some videos. So there's not a whole lot of like real close-ups of the work because I was filming it on by myself with a tripod. But it's something, if you guys do want something else just explained a little differently, feel free also to like, search across YouTube if you want to see better depictions of heat mixing and stuff like that. So tell you what, living by the lake, it is like bugs. Right, I don't know if I said that already, but it really is though. Yeah, I think that's it. Pretty happy with that. Okay. Alrighty, so I'm gonna call that a day. We got done early, the poor folks before you guys got dragged on a little while. But I got to shut up a little bit more with you guys because I was doing. Do you have any questions about any of this stuff? Or the exam is coming up? I will say this. I know I went over the exam, like an example of a critical thinking type of a question that you might see on the exam. Or like put you in a scenario and then ask you a question about what happened in it. They're not all like that. Some of them are easy questions, definition based. But to review for the exam, I typically go over a Kahoot, and it's a lot of questions that are similar to what we see in the Kahoot. Yeah. So it's a lot of things that I would see similar to what the stuff we have in other Kahoots. So there's not a whole lot. I can't put critical thinking questions in the Kahoots. I'm limited by how many words I can put in it, but it's great for studying your basically recall. So at least there is that, and we'll probably do a mini. I say a mini review, and I don't mean it like that, but I will just remind of the topics that were covered in each of the like bullet points on the study sheet. And then briefly, and then we will do like a group team kind of collaborative game on Kahoot that actually works really well for like repetition and recalling and everything like that, which if you have a limited amount of time for studying, it is good for that. So we'll do that. That will be on Monday evening, probably around 8 o'clock. I'll send out a reminder about it on probably on Monday or maybe over this weekend. I don't know, but I just feel like absolute crap right now. So I haven't done it today. So, but yeah. So for studying for the exam, definitely I would start with what I think is like the basics of making flashcards of the information on the study sheets. That study sheet for Unit 1, I'd go over that and make flashcards for it. If you guys are watching the lectures, you really need to be watching the lectures. Just going through the Kahoots, it's only 10 questions, y'all. That doesn't even come close to covering the information in the lecture. If you're just doing the Kahoots as the activity and then the quizzes and then kind of sorting through your notes for that and hoping that'll get you through the test, you will not do well. A lot of people do that and think that that's going to be fine, but it will not, because I expect you to understand it, not just like vaguely remember something you saw. So I just recommend making at the very, very least flashcards from the information on the study sheet. Yeah, I know it's a lot of information, but it's way less than what's in the textbook. So do that, start there. If you are writing stuff on your flashcards and you're starting to identify terms or references or something that you don't quite understand, or you realize that it's a reference to a concept that maybe you need to spend more time on, go make more flashcards for those things and add them into the pile and start there. And then I would go moving through with like, go through your flashcards. If you know it, if you really, really, really know it, don't need a flashcard for it. You can get rid of it if you already made it. Don't waste your time looking at it ever again. Go through your flashcards, put them in pile 2, and work to try to get things in pile 2. But if you're not confident about it, keep it in pile 1. Come back to it the next day and go over it again. Then in pile 2, go through that. If you knew it, the next time that you're seeing it, if you knew it, it goes in piles 3. If you didn't know it, it goes in pile 4, and then continue with it like that. And as you get to pile 3, that's the one that I would leave as a single pile. You don't even need to touch it until the day of the exam, probably right before the exam. Just go through and remind yourself of those terms because you already knew them. You don't need to kill yourself with it. You don't need to familiarize yourself with the wording on it. You need to familiarize yourself with the actual meaning of the information on it, right? So another thing that you can do if you want to take it to the next level, that's the bare minimum is the flashcards. Bare minimum. The thing that I would do to actually make sure you understand that material is either re-explain the material to somebody else or create your own questions with it, like I try to do with the extra credit. I try to force you guys to do that, but some of you guys are doing pretty good with that. Stuff to reformulate the thoughts into different forms. Don't keep it in the words that are on the study sheet. Formulate it somehow else. Explaining it to somebody or something else, even explaining it, hello, gold lip gloss. Would you, let me tell you about metabolism today, and all that. So yeah, I don't know. I would probably spend my time trying to do that, more than I would focusing on other things. Flashcards, and that is the best way, I feel like, to really spend your time. Especially if you feel like, okay, well, I don't understand about viruses very well. That's the one you go explain about. If you understood about chemistry, you don't need to be explaining about the chemistry part. Go explain about things you don't understand. And yes, that means going back and looking up some of the information and trying to figure it out. Pretend like you're presenting it to your colleagues, to your peers, to me. And that's how I would spend my time studying if it was me. Get the basics with the flashcards and then go on to the explaining part. Yeah, it's a lot of work, but I mean, that's college. So anything else that you guys have any questions about? Or anything I can help with while you're studying? I hate leaving people to the lecture. I hate that. I hate it, but it is what it is. I'm sure you guys are doing wonderfully with it. Nothing else? Okay, if you guys have any questions as you are studying, of course, you can always email me, and then I will be available, of course, on that review day probably, like I said, Monday at like 8 p.m. because I know people wait to study, and I feel like Monday at 8 p.m. isn't too late to have it, but it waits until it's late, till people have actually had a chance to study the material. So I'm hoping that is when we can have a good productive study review time. It's really hard on these 8-week courses. Yeah. All right. Everybody stressed out? Me too. Yes, quiz question. I said first to arrangement, and one was Staffel and the other was Palisades. Okay, so Staffelo is just random bunching, and then Palisades is, that's a good question, and it's a good way to here to help clarify it, right? So so Staffelo, you'll just have random bunching all over the place, and Palisades, it's like they stack up next to each other. So actually, now that I'm here, and I can actually just do this for you. Let's look up Palisade. Imagery. I'm hoping to see some pictures and not just drawings. You just never see them. Here's a good one. You see how they're kind of stacked up next to each other like that? Those are Palisades. That's a pretty classic image. Whenever I was showing you guys about Staphylococcus, it's just kind of bunched up like that. Yeah, just random bunching. And we will not see Staphylo. Here's another identifier. You will not see Staphylo arrangements for bacilli, and you will not see Palisades for cocci. So Palisades are only for bacilli, for rods. That's why they're like fence posts. So you can't have fence posts if they're round, right? And so they're kind of, think of Staphylo as being like Palisades, but one is random bunching and the other is like fence posts, and they're fence posts next to each other, and that's the more important part than them being like all over the place, if that makes sense. Does that help a little bit? I hope. Anyways, it's good identifying. Let me see what the heck I was even doing. Okay. Anyways, anybody have any other questions? Oh, I get what you're saying. I get what you're asking. Okay. I get what you're asking. Get the question. Yeah. I get it. So you were asking, so the question had something to do with arrangement specifically, and then two choices, and there was Staphylo and there was Palisade. Yes, they are both arrangements. I guess I just explained the differences for no reason. I'll have to look it up because I think the questions are going to be different for everybody. And I don't want to look it up here because there might be people who haven't taken the quiz yet, right? So, I could quit screen sharing, I guess, but then you see my face all close up, and then I could look it up. We have a minute, so I'll just go ahead and do that so I can get you your answer while you're here. Let me stop screen sharing. I guess, maybe not. How do I not? I don't remember stop screen sharing before. Stop share. There we go. Okay, so you guys are still there, right? Just looking at my face. Face up. All right, let's see. And that would have been chapter 3. Because sometimes if it's wrong, if I put the wrong answer, then I put the wrong answer. But we'll see if it is. It's been around for a little bit, so most of them have been worked through pretty well. Was it in the lab quiz? Let's see in the chat room. was the chapter, was it chapter 3? For some reason, I can't find it. Differential studying. Oh, chapter 4, okay, that kind of makes sense too. They kind of have a little bit of overlap. My bad. I want to find chapter 4. Sorry, I know this is boring. If you guys don't want to hang around for this, you don't have to, we're done. So there might be questions that people might have that you miss. Yeah, you're right. It does say that. I'm gonna change that. It won't, I don't know if I can't change it and like change, like your grade for it. I can give you a point for it, but like, I can't like change the answer and then it automatically gives you if you're right or wrong is what I'm saying. So, depending on who's taking it already, but yeah, good call. When people don't tell me about stuff like that, I just don't know about it. British is a term that refers to bacterial cell arrangement, yeah. Strepto. I'm gonna change that. All right, anyways. Anybody else have any other questions? All right, guys. That's it. That's all I got. So let's get out of here. I'll see you guys on Monday. I will be available for you guys to contact, of course, anytime. If you guys email me, I get back to you as soon as I can, even through the weekends. So don't worry about that. So as you're studying, please feel free to contact me if you have questions. And if you need to discuss something, you can always ask me to get onto Zoom and I'll try to make an appointment or whatever. Say, oh, I'm at the grocery store, I can't do it. But like, that's one thing. But if I can't, then I'll make a time to do it with you. So just let me know as you're studying. And I'll help out when I can. All right. That's it. I'm gonna go ahead and kick you guys out and